This is a video lecture for microbiology for Tuesday, March 31st. And today we are going to finish chapter 24. We've been talking about environmental microbiology, and first of all, I want to check the audio. I'm going to turn it up a little bit. I am thinking that you can't hear me very well. Let's see. I'll see if I can make this a little better for you. Also, I sent out a reminder that we do have a test next Tuesday, and the test will be on Moodle. You are on your honor to do it without your book or your notes, and it will be 50 questions, multiple choice true false only. So anyhow, be prepared for that on Thursday. I will do a study session. The study session will be a video lecture uploaded on YouTube, and then I will also be available for questions via Zoom or via cell phone during my office hour on Thursday. So if you have any questions or anything comes up while you're studying for the exam, please let me know. I also posted a study guide on Moodle for the 50 questions. So make sure that you start to go through that study guide and that you've gone through it completely before I post my review on Thursday. So today we're talking about monitored natural attenuation. We're basically going to discuss some different bioremediation methods that are available. And some of these methods are more cost effective, less expensive, and one of the least expensive is monitored natural attenuation. So what you're doing here is you're looking at some particular contaminated sites where the soil nutrients alone, what's already in the soil, will support contaminant degradation. And this is often the case with things like gas stations where there are soil nutrients and there are soil microbes that will degrade the gas directly. And so if you monitor the site and you don't do anything, you don't recirculate the groundwater or anything like that, then you can just by monitoring the progress, then see how long it takes in order to degrade the contaminant. So you could let the site sit for a number of months, maybe even years, and then see if the contaminants are degrading over time. Okay. If you can prove that a site is going to clean up itself, then the EPA may approve monitored natural attenuation as that method for site cleanup. That's basically a license to monitor what's going on, but in terms of active remediation, you're doing nothing. You're not really doing anything. However, a monitored natural attenuation site is still very active because you are taking periodic samples of the groundwater and the soil and making sure that the contaminant is being degraded over time. So you need to make sure that the contaminants will be remediated, especially before they impact human health or the environment. So you want to make sure that if somebody has a well where they're getting drinking water, or if there's a lake or a river involved, you have to make sure that the site is cleaned up before the groundwater hits the well or the lake. Okay, and here's an example of monitored natural attenuation. So you have these barrels or drums, 55-gallon drums of perchloroethylene. There's about 10 drums, and that constitutes about 2,700 kilograms of perchloroethylene that leaks through the soil into the groundwater. Okay, and then what you monitor is the progress of this remediation as the contaminant flows further and further away from the source. Okay, so the first thing you would want to do, obviously, you want to clean up the source, but then you want to monitor this and say if you have some downstream wells here, then you want to make sure that by the time the contaminant plume hits these wells, that the contaminant has been completely degraded. So as this water flows, and it flows very slowly, just as a matter of inches per day, As this water flows, then there are microbes that will naturally take care of it in the soil, and you just put monitoring wells downstream here, more monitoring wells downstream here, and you make sure they say if there's an exposure like a river or a lake or a municipal water supply, you make sure that the contaminant is completely degraded by the time it hits these wells here. Okay, another method that's much more aggressive and much more expensive is called bio augmentation. And this is when you add organisms, you add microbes directly to the contamination, and the microbes have been suited and grown up in the laboratory or in some facility in order to degrade the contaminant directly. Okay, so you add the organisms. You can add them by pumping them out into wells. You can add them by spraying them onto surface contamination. Okay, and so a lot of times this is what happens with oil spills, especially when oil spills wash up along the shores of, for example, the Gulf of Mexico or the Gulf of Alaska. And so what happens are bacteria like E. coli are just sprayed directly onto the surface. And then in some cases, you may have more specialized bacteria that would go into the subsurface, especially for the degradation of perchloroethylene. Perchloroethylene degrading bacteria have actually been isolated from sewage sludge, but you don't really find them very much in the environment, so you have to add the bacteria directly. Here's an example. This was from the Exxon Valdez spill, and you can see that these individuals that are suited up, they're being protected from the microbes, are spraying the microbes directly on the contaminated site. And that petroleum is very, very sticky, but over a period of days, then the microbes, which are basically E. coli, then will degrade the contaminant. Now, there's a lot of snake oil in this particular industry. Everybody has their own favorite microorganism that is supposed to take care of different types of contaminants. And so, but there are many different aerobic bacterial strains that will consume crude oil. So you could probably just pick one. If you had a massive level of surface contamination of crude oil, you could probably pick one and it would work just fine. Okay. Most bacteria, once the crude oil is gone, then they won't persist in the environment. So as the contaminant is their food source. So as soon as the contaminant is completely degraded, then the bacteria will die. The bacteria also must be shown to be GRAS. GRAS stands for generally recognized as safe. And so they have to be tested in the laboratory. And I've actually tested some of these formulations in the laboratory to make sure that they were safe and that they were not going to pose a hazard to workers and not introduce a new hazard into the environment. Now, here's an example of bioaugmentation that was done in the early 2000s. This is where there was a field that was contaminated with perchloroethylene and trichloroethylene, and they added microbes directly from sewage sludge that would degrade the perchloroethylene under anaerobic conditions. Okay, this was done at Kelly Air Force Base in Texas. A lot of times Air Force bases are a prime candidate for contamination because they do a lot of washing and cleaning of aircraft parts. And they use a lot of jet fuel, and both of those are persistent contaminants in the environment, so they'll need to be remediated. And the organisms here were actually dehalo-respirating microorganisms. They actually breathe chlorine. So they use the chlorinated compounds as their terminal electron acceptor, or we could say just roughly they breathe chlorine. And the microorganisms gained energy from this dechlorination process, so it was energetically favorable that this would happen over a period of time. Now, you have to be really, really careful with this type of degradation because you start out with perchloroethylene. That's basically ethylene with 4 chlorine molecules. You dechlorinate it, it goes to trichloroethylene. You dechlorinate it further, it goes to dichloroethylene. You take off the second to the last chlorine, it becomes vinyl chloride. And vinyl chloride is probably the biggest hazard here. So even though you've started out with a contaminant of perchloroethylene, as you're decontaminating, you want to make sure that you don't accumulate vinyl chloride because it's a potent carcinogen. And then vinyl chloride, once it's dechlorinated, becomes ethylene, and then that's just released in the environment. It's degraded by natural microorganisms. Okay, with the Kelly Air Force Base, when investigations were done, they found perchloroethylene, trichloroethylene, and dichloroethylene in the groundwater. Okay, and the groundwater was generally aerobic, but they thought that in the microscale, there was reduced manganese, which would suggest anaerobic activity. There was also significant levels of nitrate and sulfate in the groundwater. So this means that not only could you stimulate aerobic organisms, but you could also look at manganese reducing organisms, denitrifiers, and sulfate reducers. Okay, and this is how the injection occurred. Okay, you had a nutrient delivery well here, and through the nutrient delivery well, you would also introduce microorganisms. And then you would have monitoring wells that would go with the gradient of the groundwater. The groundwater would naturally flow here. And then you would have an extraction well that would just recirculate the water and go back into the nutrient delivery well. So you'd have a recirculation system. You have a cone of depression here because you're taking out groundwater, and then you have a cone of expansion here, this cone of expansion, because you're re-adding groundwater with nutrients. Okay. And simply at the very beginning, all they did was recirculate the groundwater for 89 days. And just by stirring up nutrients in the soil and in the groundwater, then that was enough to start to degrade the contaminant. So you did see some contaminant degradation. Then at 89 days, nutrient was added. And when nutrient added, then that bolstered the existing microbes to further dechlorinate perchloroethylene. Groundwater, like I said, was removed from the extraction well, and then it was amended with carbon sources, methanol and acetate, that's just methanol and vinegar, and re-injected into the injection well. And then after about 176 days of operation, then they added dechlorinating bacteria, a strain of bacteria called Dihalococoides athenagenes. And this would basically breathe the chlorinated compounds and selectively dechlorinate the compounds over a period of time. And you can see what happened was that there wasn't a whole lot of monitoring and sampling that took place here, but you can see this top line is perchloroethylene, and this was in micromoles per liter. You can see an abundance here at the very beginning, but just recirculating the groundwater, then you started to see perchloroethylene and dichloroethylene both go away. Okay, there was very little trichloroethylene that accumulated in a process and almost no vinyl chloride. So just by recirculating the wells and not adding nutrient, then you saw a decline in the concentration of contaminants. Then once they added nutrients, this was methanol and acetate, and then they saw perchloroethylene was a little spurious, it went up. That just may be little pockets of contamination that were at higher concentrations. But throughout the remediation, then they went back down, and you got an accumulation of dichloroethylene. And then by the time you added the organisms, then dichloroethylene then ultimately was degraded. And you did see some increase in vinyl chloride, and we want to be very careful with the increases in vinyl chloride because that's a more potent carcinogen, and that's a much more hazardous to humans. So you saw that over time there were some increases in vinyl chloride, but then ultimately at the end of the remediation, then you saw everything was primarily just dechlorinated until you had ethene. Okay, so it was successful. Okay, the presence of the bacteria, there were some naysayers that said that the bacteria would just stick to the well and it wouldn't grow in the formation. But the presence of this bacteria did correlate with the appearance of vinyl chloride and ethylene. And by the end of the study, in all the monitoring wells, we saw that the bacteria broke through. So it was actually being delivered throughout the groundwater. But it was not seen outside the test area, and that was very important because we want to make sure that we didn't have this exotic bacteria get into the environment. We wanted it only in the test area, and then we want to monitor to make sure that after the remediation was done, that it just died. Now, there are still some issues with bioaugmentation. Obviously, it's very expensive, and that can be difficult. The organisms have to be grown off site. So, and often you will take your organisms that you've grown off site, and the natural organisms will outcompete them. So you'll have an unsuccessful remediation. You'll basically just be feeding the natural organisms, and the ones that you add exogenously then will not grow. Okay. Also, a lot of organisms form biofilms and they're sticky. And so when you're trying to deliver them using wells out into the soil, then they may stick directly to the well casing and just form a biofilm and plug the well. Okay, and also you have to look at what you're getting. A lot of times you're isolating organisms from sewage sludge, and these bacteria may pose a threat to public health and the environment. So you need to make sure that the exposure has been tested and also that the exposure is contained within the test zone. Okay, so we're going to finish up this chapter and we're going to talk a little bit about aquatic microbiology. So this is very important when you're testing aquifers. This also gets into food safety and microbiology when you're looking at water that is being used for food processing. And so it's important to look at all of the different dynamics in terms of water, both in the air, in surface water, and then also in groundwater. Okay, so from trees, we have transpiration, then we have precipitation, which delivers water down from the air into the water. Then you have evaporation, and then over ocean or over different aquifers, and you have evaporation and precipitation. And then you finally have runoff and then seepage of surface water into groundwater. Okay, in saltwater environments, Craig Venter, who is the person who sequenced the human genome, has discovered 1,800 new species of different organisms, microorganisms, and has done genomics for about 1.2 million new genes. And one of the bacteria that Venter is focused on is cyanobacteria. These are photosynthetic bacteria that fix carbon dioxide, and so they actually will work against climate change. And these genes are actually not a part of the bacteria themselves; they're a part of phage, and the phage infect the bacteria, and the cyanobacteria are photosynthetic. There's also other types of bacteria called green sulfur photosynthetic bacteria. They use carbon dioxide, but they don't necessarily produce oxygen. And you find these in deep sea vents where they harvest light from chemical reactions or from just the friction of breaking mineral crystals or bubble formation. And they will harvest the light and then they will be able to use that light in order to convert CO2 to glucose. Here's cyanobacteria. This is a chain of cyanobacteria. You can see that it is green because it's photosynthetic. It does not have chloroplast, but it does have thylakoids. Okay, and here is an example of photosynthetic bacteria, and you can see the thylakoids. They're single membrane structures, so there's not a double membrane, so technically they're not an organelle. You can also see the nucleoid here. You can see some nuclear material, but it's not encased in a nucleus because this is a prokaryotic organism. Some novel activity has been seen in bacteria discovered called cyanothece. Cyanothece can both fix nitrogen and photosynthesize, which is unique. You don't find photosynthesis in organisms that are nitrogen fixers. And the nitrogen fixation and the photosynthesis are 12 hours out of phase. And so what the organism has to do is to recreate its photosynthetic activity every morning. and then abolish it and then recreate its nitrogen fixing activity at night. Okay, and here's cyanotheke. You can see that it's green, and so it's in its light phase. It's actually photosynthetic. And what it does is in light reactions, it can take carbon dioxide, convert it to glycogen, and then use that glycogen to fix nitrogen during the night. Okay, and here's the overall reaction. Don't worry about that too much, but it basically, the sunlight reactions, the light reactions accumulate ATP, and then the ATP then can take nitrogen and convert it into ammonia. It's actually ammonification, and when that happens, then that provides a nitrogen source for the organism. Now, just switching gears a little bit, aquatic communities are important, and you do get animals that comprise plankton, and these are in the upper portion because they do need oxygen. So you have phytoplankton, which is more plant life, and zooplankton, which is more animal life. And then you get aerobic bacteria, and then below that, anaerobic bacteria. Okay, and these bacteria support lower invertebrates and fish in the community, and so in the lower levels of the aquifer, then you'll find these bacteria, and they provide a food source for the food web. Here's an assortment of plankton. You can see phytoplankton and zooplankton. Okay, I'm sorry, let me go up. This is phytoplankton only. And then we have zooplankton here, which are more animal life, and they're part of the animal kingdom. And you can see more complex structures. Now, during the summer, you get what's called thermal stratification. And so the colder water will sink, the warmer water will be at the top. Okay, it will rise, and you have the epilimnion, the thermocline, and the hypolimnion are the different phases of the water. And then in the spring and fall, then you get seasonal upwelling because of nutrients creating heat at the very bottom of the aquifer. And once those nutrients then create heat, then the water rises, and that creates a current. Upwelling is one of the explanations for something called red tide. Red tide. Red tide is a type of algae, and the algae is a dinoflagellate algae. It produces a toxin, and red tide is consumed by shellfish, and so this is a paralytic toxin. And so you see this in the spring and the fall, where there's more upwelling, where there's more nutrition that's being recycled from the bottom of the ocean up to the top. This is what red tide looks like. It's very intensely red, and these are the individual organisms. Here, Carina brevis. It's also important to maintain oxygen balance in an aquifer. Okay, oxygen is not very soluble in water. Its solubility is very low, so you only see it at the very highest levels of water. Okay, it's going to be highest in the epilimnion. That's the layer at the top that's warmest, and lowest at the bottom. And most aquifers are oligotrophic, meaning that they have very, very few nutrients. The nutrients are being scavenged as part of the food web, and so it's nutrient deficient. Okay. Now, if an excess of nutrient is added, this is called eutrophication. And what happens to the aquifer is, with excess nutrients, then you get heavy surface growth of algae and cyanobacteria. But what becomes a problem is that shuts down the oxygen supply to the lake below. So even though there's a lot of nutrition there, the oxygen is being consumed up in the epilimnion, epilimnion, and that is being consumed by algae and cyanobacteria. That depletes the oxygen, and it does not allow the oxygen to diffuse to the lower layers of the aquifer, and it shuts down the oxygen supply. So it looks something like this. This is eutrophication, where you just see a big, big green haze at the very, very top of the aquifer. And you know that fish below that are oxygen starved, so they'll start to die. Okay. You can see a normal lake. The normal lake to the left is a oligotrophic. Okay, so there's oxygen and nutrition to go around to support the food web. But then during eutrophication on the right, then you get a big algae bloom. And when that happens, then below there is no oxygen. The fish will starve or will suffocate because of the lack of oxygen. Okay, and that ends this particular lecture for chapter 24. I will continue on, and there will be a lecture for chapter 25. And after that lecture has completed, then I will post a review lecture for the exam.